First of all, nice to meet you and welcome. My name's Kevin. I'm the president and part of the exec team here at SynBio Canada, and very excited to see you all joining. In case you're not too familiar with what SynBio Canada is, we are an organization that is focused on uh, broadly on community building in the synthetic biology space across Canada. And that includes like making connections between all kinds of stakeholders that are interested in SynBio, but also working on seeing what opportunities we can bring for students or what kinds of funding we can generate towards this interesting field that we're also passionate about. We are a nonprofit and entirely volunteer run for now, and we're very new to the scene. I feel like we're still building a lot of our teams and communities, and can always use more volunteers, and are slowly expanding what kinds of activities we want to do. So this is the first. It feels like a first event, at least for this year, and a good little like moment of connection between all of us. And it's so exciting to see so many people that have registered and that are joining. This is so fun. Um, okay, I think a couple other things to clarify. So feel free to use the chat as much as you want during the event. Let's keep that lively, and good to see everyone. Good to see you too, Luana. Um, and we are recording the presentation portion of the event, and we'll try and post that to our YouTube. Hopefully that works out and people can take a look at what's up there. But we'll turn off the recording for the Q&A portion of the event. Um, and I think that covers everything. At the hour, we'll switch from presentation mode over to networking breakouts. So we'll get into little groups of people. You can actually get to meet some of the people who are on this call, as opposed to just tuning in and tuning out. We'll try and give ourselves a moment to make a few new friends, and I'm looking forward to that too. Um, and I think I've covered everything. Lee, is that good? Can I pass it to you now?
Yeah, for sure. We can get started.
Awesome.
Um, yeah, so I'll give a brief introduction to our two panel speakers today, who we're very thrilled to have with us. Dr. Scott Pownall is the president and co-founder of the Community Biolab Open Science Network Society in Vancouver. With over 35 years of experience spanning academia, research institutes, and industry, his passion for exploring the world through SynBio has really driven his work engineering organisms from microbes to mammals. Scott is also a strong advocate for open science, having developed tools like the Open Yeast Collection, and he's collaborated with other initiatives like the UK-based Open Bioeconomy Lab and the Reclone Network. He also serves on the iGEM engineering committee, so he's done a lot within the community to advance accessibility in SynBio. We also have with us today Dr. Benjamin Scott, who leads the engineering biology platform at the Global Institute for Food Security, where his team harnesses lab automation to achieve new scales of agriculture and agri-food product development. Ben's expertise in protein and genome engineering has been applied to projects spanning medicine, agriculture, and industry. And back in 2018, he actually founded SynBio Canada, which is the not-for-profit organization we're all a part of, which unites researchers and showcases talent. And so that has ultimately brought all of us here together today. So we're very excited to have both Ben and Scott here to kick off the very first SynBio Canada event of the year. And with that, I will leave the floor to you, Ben and Scott.
Thanks so much, Lee. Really delighted to be here, and I think it really timely presentation as well, thinking about building something special here in Canada. And Scott and I have put a lot of thought into that over the past year, believe it or not, and we're delighted to present kind of some of our thoughts and talk about that with the community today. So what is the bioeconomy? Well, my favorite definition is that it is jobs, and everyone wants a job. All, we need jobs in Canada, and politicians love talking about jobs. So whenever you think about building a bioeconomy, it really means building secure jobs here in Canada. And those jobs are nonprofit as well as for profit. And these are both needed, they're not exclusive, and they both fulfill really important aspects of the bioeconomy. And we need both here in Canada as well. So Very simple, straightforward definition for any non-scientist, for any sort of policy advocate, or just getting introduced to the bioeconomy. Think of it this way. A little bit more detail here when it comes to both investment as well as what it takes to go from a discovery to a commercialization. There's a lot of steps involved and different ways of categorizing them. My favorite way is to explore this in sort of two ways. One, which is discovery driven research, which is typically the type of research that occurs in academia. And Canada is actually being very good at investing in this. We've built great talent as a result. But then getting that to a commercial product, there's what could be characterized as a valley of death in between these two spots. So what typically happens, and what many of us have already experienced, is that you'll discover something wonderful in your lab, you'll create a groundbreaking publication, and then what? And it's really hard to take it beyond that. And that's not any one person's fault. It's systemic to the way that innovation works. With industry money in particular, Canada is not doing such a good job. So although we've built a lot of great talent and we've made incredible discoveries at the discovery level, Canada's private R&D has been lagging for decades. And of all of these countries here, so these are all countries that we're competing with. Canada is the only one where year over year R&D private spending has gone down, and particularly since 2020. So how can we make Canada's bioeconomy more competitive? So obviously, the answer would be we need to invest more, but it's a little bit more complicated than that. Competitiveness actually means we need to offer something that no one else can. And I think that's actually a more creative and I hope more inspiring way to think about what we can do differently here in Canada. And it really comes down to we have to build something unique to foster, retain, and lead the bioeconomy, which is accessible by both aspects of this bioeconomy, which are nonprofit and for-profit researchers. So we can't be doing the same thing over and over again. It's clear that the current strategy of building up great talent in universities but not investing in commercialization is not working. So let's start with building something new. And because it's top of mind for all of us, we really can't escape from the news these days. I wanted to try to reframe that as a bit of, this is an opportunity we can't escape from, rather than some really jarring and a roller coaster ride anytime you turn on the news right now. So I want to reframe that as an opportunity. I'm actually going to start a little bit back in time. So in 2022, the Biden administration, so this is in the US obviously, had an executive order on advancing biotechnology and biomanufacturing. And when I read this, and I encourage you to, if you can find it, like honestly, the American government is scrubbing a lot of this sort of stuff from the internet. But if you can find a copy of it, it's really, really impressive. It announced $2 billion in new funding, which is over twice CIHR's annual budget. And had 40 very specific objectives spread across numerous agencies. And they touted this as a whole of government approach, which is exactly what you want to see coming out of a government that has so many different branches. You want them all kind of working together. And some of my favorites, because I think they will resonate with the community here particularly, is that these objectives included expanding biotechnology training and education, a strategy for expanding domestic biomanufacturing, a unified website for biotechnology regulation. So just like where to go to get information about how biotech is regulated. That's not intuitive to many people. So having one place to go is really important. And then last but not least, a biosafety and biosecurity innovation initiative, which was actually specific to how to screen and produce safe and secure synthetic DNA, which is going to immediately relevant to this community and what Scott and I are going to talk about today as well. So you can actually read up more about all of these different objectives on this nonprofit website that summarized these different objectives. It's a great read for any policy or anyone interested in bioeconomy. But then the end of the story, or the where we are now, is that this executive order was rescinded 2 weeks ago, along with 18 others by the Trump administration, and it went with not a lot of fanfare, not a lot of people noticing, because of just the deluge of information now. But this is devastating for the American bioeconomy. And what I want to reframe that as is, and rather than rehashing everything that's in there, that I want to argue that this playbook is now there for the taking, and we should seriously explore as a community and as a nation what we can learn from this and how we can take this and run with it, since the Americans clearly aren't interested. So that's one aspect that I'll leave you with. But then I want to explore, well, what does Canada already have? So we're not starting from scratch. So what do we have already when it comes to the bioeconomy? And one thing that typically comes up in any conversation about, well, what does Canada have? What is the Canadian economy built on? It comes to national resources. So these are resources that we have here in Canada that are, oil, timber, gold, stuff you dig out of the ground. But there's other resources that I'm going to talk about that are a little bit different. So what are other examples of Canadian national resources? So there's one that you may be familiar with and you may not have ever heard of, which is the Digital Research Alliance of Canada. And this is a screen cap from their website. They have a beautiful website, and I encourage you to go explore it as well. It essentially means that Canada has supported 5 supercomputers at universities across Canada, and they're located at universities in BC, Ontario, and Quebec. And these supercomputers offer advanced research computing. So any researcher, academic researchers and for-profit researchers can access this research computing. So they don't need to buy and build their own supercomputer. They also offer data storage and research software. And this was all supported by a direct federal investment of several $100 million spread over a few years. And since then, because of this interest in AI and the way that technology has developed since the Digital Research Alliance was founded, it's led to additional add-on investments. So once this was set up, it attracted even more money because of the utility for the Canadian research community. And so if you are thinking about needing a GPU cluster, CPU cluster, using, you're dipping your toes into using a supercomputer, you can do that relatively easily thanks to the Digital Research Alliance. Another example of a Canadian national resource, so Genome Canada, although they may not have physical research resources, for the exception of Genome Quebec, I will argue that Genome Canada has been foundational in ensuring that genome sciences and now biotechnology and engineering biology is getting direct investment. And they have a over 25-year history now of $4 billion invested, and this is not insignificant. And they have a very unique structure as well. This is probably more for the benefit of people getting introduced to synthetic biology, but I do want to point out just how foundational Genome Canada is in Canada and how unique it is as well. Okay, so now I'm going to get a little bit more technical. That was the policy stuff, and since some of you are wet lab scientists, you might appreciate this, but I'm going to go through this in a little bit of detail for those who aren't. So in biotechnology, there's essentially an iterative cycle that we can complete. And there's 4 components to that are outlined here. And I borrowed this from a publication. There's design, and that primarily means you're planning out how you're going to engineer your organism. This will include identifying what DNA sequences you're working with and how they'll be assembled and put into your cell. But eventually you need to, leave that work behind in the computer and apply it in the real world. And that requires the next 2 steps, which are build and test. And this needs physical components. So you can't do experiments without, chemicals, media, cells, and then ways to measure their activity as well. And without, a perfect model of the way a cell will work in the computer, there will never be a way to escape from this. And then once you do generate all of that data, Learn is the last aspect. And obviously, as humans, we're able to learn from any finding that we get. But what this really means is applying tools to understand the very large data sets that we can now generate in engineering biology. And if you're fortunate or able, you may use lab automation to speed up certainly the middle two processes here by automating repetitive tasks. And the cycle is iterative. So once you've learned from your round of experiments, you'll go back and design more and conduct another round. So what I want to argue and what I want to, for you to think about is that the last two aspects, or the first and last, design and learn, are actually becoming far less resource and time intensive. And this is generally thanks to advances in domestic computing power. So we don't need supercomputers for most of what we do. And a laptop is sufficient for running the sort of design and even some learn aspects. And now there's machine learning algorithms, which, if you're skilled in using them and you feed them good quality data, they can be transformational and speeding up this cycle. But the middle two aspects, so build and test, remain very time intensive, and they continue to bottleneck innovation. And that's even with lab automation. So I'm a huge proponent of lab automation. I have some experience in using it and applying it in my own lab. But it's not a panacea. It's not going to solve all of our problems. And it's really just the best way to perform repetitive tasks, not to necessarily come up with something new. So here we get to a little bit closer to the crux of things, which is that a national resource that makes build and test faster would directly accelerate the bioeconomy, primarily because these are the two hardest steps. Maybe not hardest, but certainly the most time intensive. And those, therefore, an investment in supporting how quickly we can complete these will be transformational for the bioeconomy. So build and test require physical components, and we can't escape from that. And that means two major things: synthetic DNA and chassis organisms. So synthetic DNA means DNA sequences that don't exist in nature. They might be inspired from nature, but they aren't coming from an organism typically. And then chassis organisms. So this means cells, strains, animals, plants that we can use to engineer for whatever purpose is required for the goal. And both of these, except with the exception of maybe higher organisms like animals and plants, they're relatively cheap to store and replicate, but they're expensive to create, which essentially means that once we've built them, you can propagate them very easily, but it's the initial time and money investment that is the biggest hurdle for creating these two things. And then with lab automation, it certainly unlocks a brand new scale of research, but it's also expensive to obtain, operate, and maintain. So if we're going to focus on these aspects, a national resource that provides easy access to these physical components, so these things that exist in the real world, not in a computer, will get us to a faster build and test cycle. Before I move on, I wanted to bring up this idea of intellectual property. And I'm not an IP lawyer, but I do think that for the context of this talk, there's a few really important definitions that Scott and I are going to refer to. So one is patents. And what a patent means, and when I was in grad school, I knew patents were important and that the patents meant money, but I didn't really understand what it really meant. And now that I'm a little bit further in my career, this is my favorite definition. So what a patent is, it means you own the right to make money off of your invention for a limited period of time in exchange for providing an enabling disclosure of it. And what this means in the context of our talk here today is that patents can block the use of specific DNA sequences and chassis. So it essentially reserves the right for the person with that patent to make money off of that until the patent has expired or perhaps challenged. Another type of intellectual property are called material transfer agreements. And these are not patents, but they do enable you to control who has access to this physical material and what this material can be used for and how it can be shared. So this is typically in academia, you may have come across the Uniform Biological MTA or UBMTA, particularly when you're using AdGene to obtain a plasmid or a strain maybe from ATCC. And these provide standard terms, but it prohibits commercial uses and you can't share it with anyone else. So it's actually very restrictive when we want to think about moving beyond academic research into commercializing something. Now, if something isn't patented, you can just synthesize it and you don't need an MTA in order to use it. Now, again, I'm not an IP lawyer, so you need to ensure that you're following all the rules and that it is not patented. But if something isn't patented and you are interested in using it, you can just synthesize it. So why not make this DNA open? And to put it another way, imagine you go to AdGene or you see a DNA sequence published in a paper. You do your due diligence and ensure that there's not a patent on it. But how do you actually access it? So there's really two ways. One would be to ask the researcher, and one would be to synthesize it yourself. If you ask the researcher, you're typically going to be sent down a rabbit hole of emails. You might be fortunate enough to get a license for it or an MTA, but then you can't use it for anything beyond academic work. So why not just invest a little bit of money to make that open for yourself? But that still requires you to make that investment. All right. So this inability to access synthetic DNA and chassis organisms, I'm going to argue, directly contributes to this valley of death, where we have a wealth of strains and DNA and cell lines that are sitting in freezers across Canada, and they're not getting used for anything. And they may not have any patent, but there's definitely going to be MTAs associated with them because these institutes are not going to release them freely, typically is the current way of doing things. And as a bit of a joke, but I honestly kind of have a nightmare about this, where I like my strains and plasmids that I built in my PhD and master's just never see any other use, and they just sit languishing in this valley of death in a freezer like one of these, and that's the end of the story. And I really think that's a shame and something that we can actually change. These resources exist, and we should be looking for ways to use them. Okay, and I'm going to turn it over to Scott now, who will.
Yeah, thanks, Ben. That was a great setup, and yeah. So we've been talking about what approaches can we take to actually promote innovation. And for me, coming from the community lab, which is right outside of the academic arena, I was looking at the Valley of Death image. There are a lot of young people that are getting jaded with the academic life, and because of the pyramid of trying to get further up and up to be able to do basic science, they've turned to trying to start up their ideas. So how do we actually reduce those barriers? I would argue that for those people, the beginning of that TLR graph is flattened down along. So the Valley of Death is really from the beginning onwards. How do we get that bootstrap? And I want to bring in this publication that came out back in October of 2018. This is by my collaborators, Jana Malloy, Drew Endy, and Linda Call, who was the senior legal advisor for the BioBricks Foundation that Drew Endy founded. And those people that are familiar with or are into iGEMs are familiar that Drew Endy was one of the founders of the iGEMs competition. But they created this open MTA. So it's an alternative to the Uniform Biological Material Transfer Agreement, which is highly restrictive. So I'm just going to run through the 5 characteristics of the open MTA. The important thing is access. Access is free of any royalty or fees. You can actually charge a transfer fee, something to cover costs. Attribution, providers may request, so it's up to them to decide whether they want acknowledgement on the resources that they supply, for a publication or whatever. So it caters to both academics and people outside of academia. Reuse, you can do what you want with the materials, whether it's engineering it further, or using it as a part within a subsystem that you're building. It's totally up to you. The next step is the redistribution. So there are no barriers for you sharing this. And coming from community biology, this has really been a big game changer. And it's something that I've been supporting for a long time. Is you can't restrict somebody from sharing or selling. So, for example, if you've got an idea to build an educational kit, say, and you grab a bunch of parts that you put in to teach people, you can go ahead and do that. You can sell it. So there's no restrictions. Like in the software arena, we've got copyleft software licenses, and then what are called copyright software licenses, which are more permissive. The next step is non-discrimination. So basically the materials can be transferred to anybody. And I think this is really fundamental. Academics think of academia as the originator of ideas. Companies think of companies as the originator of ideas. I think of people as originators of ideas. And there's a whole lot of people out there that really are strapped. And I've had people come through my lab and build companies starting off in my low resource intensive arena. So this open MTA, I think, is really important as a starting point to be able to go and support innovation in the bioeconomy here. Next slide. So one of the things, this is a list off of the BioBricks Foundation's website of organizations that have already become signatories to it. And it's interesting that there are no Canadian universities on this list. And, I'm wondering why. But I think it's something that we need to do, is that we need to get the different organizations to become a signatory, because the Open MTA really does support innovation. Next slide. So what I'm going to talk about is, my connection with all of these toolkits really for engineering biology. And back in 2018 at the Global Community Bio Summit that was held at the MIT Media Lab, Drew Endy came and presented about the Open MTA, but also the Free Genes project that his nonprofit, the BioBricks Foundation, founded. Originally it was called the BioBricks 10,000 Gene Project, but that was a mouthful. And so Isaac Larkin and Keany Gandol coined the term free genes, and so that stuck. And the four people there, they worked to support free genes internally. Isaac was an outsider, friend of Keany. But what Drew Endy did was he got a grant or funding from a sponsor to be able to kick this off. And he collaborated with Twist Biosciences that agreed to release their materials under the Open MTA to Drew. And people could design, submit sequences to be able to have them synthesized and then distributed to anyone that asked. Next slide. I've had a long collaboration with Jenny Malloy at the University of Cambridge, and more recently she's at the runs the biomanufacturing group at the ICGEB in Italy. So she's super busy. So they created, and it's largely the work of Chiara Gandini on the left, a postdoc that was in Jenny's lab, but she's now working at Nuclera, doing amazing things there. But the open enzyme collection, the E. coli protein expression toolkits were really fundamental toolkits for engineering E. coli. I've contributed a bit to these sequences, but largely, it's Jenny's group that produced this. And this is basically a library of DNA parts that you can use to engineer E. coli. So there's a bunch of promoters, tags. So these tags are for purification or fusion proteins that, you've got chromoproteins, cleavage sites. Then with the open enzyme collection, there's a whole bunch of enzymes that people use to manipulate DNA and RNA. And then, of course, a library of terminators. So these were designed by the Open Bioeconomy and their collaborators. They're synthesized by FreeGenes, released under the Open MTA, and the work in Jenny's lab was funded by the Shuttleworth Foundation. Jenny was a Shuttleworth Fellow. And there's also other organizations that supported this work. Next slide. So that was the E. coli toolkit. FreeGenes also allowed me to create the Open Yeast Collection, which is a collection of DNA parts for engineering yeast. And that's Saccharomyces cerevisiae, as well as Commagotella fafii, commonly called Pichia pastoris, their old name. And as with the E. coli toolkits, it's free, reusable, and redistributable. It's extensible. You can create your own parts. You can use it in any aspect, from human biology, people just wanting to nurture some ideas that they have. Education, it's a great resource. I was actually at Capilano University here in Vancouver yesterday giving a talk on the educational aspects of these open toolkits. But of course, it's also very useful for research and industry. In North America, people use John Dubner's yeast toolkit, which is available under the Uniform Biological Material Transfer Agreement. So it's great within academia. Academics know about it. Not many people know about the open yeast unless they're connected with the iGEMS competition. So FreeGenes was distributing this. Drew has run out of money, so there's no new synthesis, but they also haven't, he's been working hard to try to get more funding, but I guess the current economic climate has kind of prevented that. So I think they'll keep going, but we'll see. But all of these components are released. I have many of them in my freezers here. I've transferred many to Ben at the Global Institute for Food Security. Let's go to the next slide. So this is just an overview syntax of it. So it was inspired by the yeast toolkit. And they're just a ton of parts for either episomal or integration vectors, yeast selection markers. I've now created a part to retrofit all this yeast selection markers with lock sites. It can be used for expressing a single gene, say in Pichia pastoris. I've created a bunch of new receiving vectors because the original open yeast, partly because of failures at Twist in terms of synthesis of certain parts, the receiving vectors, or some people call them destination vectors, weren't as strong. But now I've got a bunch of receiving vectors as part of this toolkit. Next slide. More recently, Jenny Malloy got a grant from the University of Cambridge, EPSRC Impact Acceleration Grant. And she opened that up to the reclone community, basically saying, much like FreeGenes, what do you want synthesized? And a lot of people submitted ideas. My lab member and colleague and friend, Ian Caven, who's a Python um, expert. He developed automation tools to take all of these ideas and convert them into actual DNA parts. So he and I worked these through the whole process to get them into Twist. And then both of these toolkits were shipped to my lab. The UK has a 21% value added tax. British Columbia only had the 5% GST. And so it was less expensive to send it to me. I replicated everything, sent it onwards to Jenny, as well as back to Drew's lab. So they're the two toolkits, the E. coli open enzyme parts, so it's an extension of the open enzyme collection, and the yeast protein expression toolkit. I'm not going to go into all the details of everything, but maybe next slide. This is just an overview of the yeast protein expression toolkit. So I take the syntax from the E. coli protein expression toolkit and many of the sensible parts that could be recoded for yeast. So I've got a bunch of signal sequences. You know, Pichia is awesome. If you can get it to secrete your protein, you've saved a lot in purification. And then there's a whole bunch of parts as fusion partners. There's even some polystyrene binding domains as part of it. But a lot of the fusion partners for purification tags, people here would be familiar with PGEX, maltose binding protein, and a whole bunch of other things. And there are also a bunch of parts that are yeast specific that go in there. Okay, next slide. Okay, so the OpenMTA is not just for DNA. Some of you might have read about the Open Pichia strain that was developed in the Netherlands. And over the past year, I've been in conversations with Katrine Clays. The title of their papers mentions license-free. It's not so license-free. There is a license. It's actually much better than the Universal Biological Material Transfer Agreement. I think I've got to drink a bit of tea. Okay, so just last week, I received from Noah Sprent, who did his PhD at Imperial College London. He looked at the Open Pichia. He was thinking about getting it, but he realized the costs were prohibitive for him. So he's just starting up his company, Change Bio, based on the work of his PhD thesis. And so he went ahead and replicated Open Pichia with one of the other NRRL strains, and he's released that under the Open MTA. And I just received it last week. I've made a bunch of glycerol freezes, and it's there to be distributed under the Open MTA. So yeah, next slide. So with this whole thing, I want everybody to think about, well, what toolkits would people want? What toolkits do people find useful? You know, there's no way that I've got a broad range of experience from engineering mice to microbes, or microbes to mice, but I don't work with plants. I've done a lot of work in mammalian cell culture, so I can design some parts there, but it's not my area. And my community lab is a biosafety level 1 lab, so most of that work would need biosafety level 2. So the question is as a community, what would everybody find useful as parts? And I think the one thing that I want to highlight is that there might be people that are resistant about the idea of having this stuff as open from the point of view of how do you exploit it. The exploitation is not in the framework. That's just a toolkit to help you get your idea translated into real commercializable science. It's really your gene of interest, gene or genes of interest, depending on whether you're building a metabolic pathway or something else. But think about not just toolkits, but also what strains. Next. Yes, okay, that's you, Ben. I think you're muted, Ben.
Sorry about that. This brings us to what Scott and I are proposing. This is a logo I made in PowerPoint, so don't think that it's the final deal here, but it is, I think, it's certainly analogous to the Digital Research Alliance in the way that we've sort of framed our thinking. So there's a few components here that build on what Scott just talked about and what he's done himself, as well as what we feel Canada could really benefit from. So part one to creating this research alliance is sharing. So relatively simple, but maybe not as simple as you think, which is the storage and distribution of open biological parts. And I'll go through each of these in more detail, but I just want to lay it out overall. Part two, build. So affordable, traceable, as well as screened synthetic DNA. So affordable means any researcher in Canada can afford it. So that means nonprofit community labs as well. Traceable means we know who it's going to and who's using it. And screened means that we have full record of what that synthetic DNA is and that it's being screened for any pathogens or pathogen-like sequences, which is common practice. But I just want to reiterate that this would be a serious component of the build. And then part three, so once we have the DNA and the strains, we need to ensure that they work. And centralizing equipment and expertise for high throughput techniques is crucial, which I'll hopefully try to illustrate. And this will get us to iterating on design, build, test, and learn faster, as well as biomanufacturing, which means scaling up what we've created. And we've outlined some organizations that we've been inspired by. And I do want to acknowledge that this idea of something open and a research program or institute set up to support this is not new. These are not new ideas. Scott and I are really standing on the shoulders of giants. But I do want to leave you with the impression, and if you leave today with one other message besides bioeconomy equals jobs, something open and accessible is achievable, and these organizations have already demonstrated that. And so I think Canada, needs to take that as an example and use that to create something here that's special. Okay, so sharing. So what would that look like? So a distributed network of open parts, and these would be research institutions, students, enthusiasts, entrepreneurs, companies, both large and small, that would be able to access them, and also perhaps act as nodes across the country, connected by a central online portal. So just like the Digital Research Alliance, there's 5 supercomputers spread across the country, but there's one website and one team that you talk to, at least initially, to set up access. And that's the first sort of core part of what this research alliance would accomplish. Thinking about national sovereignty and as well as building something unique, these reagents will be accessible by Canadian researchers and companies only, or at least exclusively for a period of time. And this is, again, this idea of creating a national resource and something special that we have here in Canada that no other nation has. What you build with these parts would remain yours, just like the way the Open MTA works. But if you choose to deposit open parts back to the network, back to this alliance, you'll gain credits for DNA synthesis. So, for example, all of those parts that I built in my master's and my PhD, if I could convince U of T to maybe deposit them, U of T would then gain credits for DNA synthesis and distribute those amongst their researchers. So there's no value in those parts currently sitting in a freezer that no one is ever going to be aware of other than myself or my old lab. But they could have value in 2 ways. One, they could be open and usable by other people. And 2, the institute that holds on to them without any knowledge of them would actually gain something valuable, which is getting credits for DNA synthesis. And a little bit more about that. So build, so that DNA synthesis aspect. And I want to reiterate this because it's really in the news every day right now. But pandemics and trade wars have plainly illustrated the fragile and actually very political nature of supply chains. And Canada actually already has a very long history of public investment in DNA sequencing. And this is acted as a type of subsidy. And I want to take that word subsidy and take a bit of time to reuse that, because I don't think that's a negative thing. This was a requirement to ensure that Canada had DNA sequencing capacity. And as I'm sure many of you have experienced, getting DNA sequencing results in Canada is actually relatively simple. There's a variety of institutes that you can choose from, and they all have many of the latest and greatest DNA sequencing techniques. This did not happen overnight, and it wasn't cheap to accomplish. This required dedicated investment. But this investment, this subsidy in developing this DNA sequencing capacity has paid dividends. So, for example, during the pandemic, this DNA sequencing capacity was immediately available for hospitals and public health networks across the country to access for COVID monitoring. And then as we think about personalized medicine, which is the way that it was framed in the initial stages, personalized medicine is actually becoming a little bit more closer to reality because of the falling costs of DNA sequencing. And that falling cost, again, is only possible by investments in infrastructure. And something else I want to leave you with, and to really drive home, is that without access to synthetic DNA, the bioeconomy cannot innovate. So we can't just be using what nature has. We don't actually even know everything in nature, obviously. We don't have access to all genomes. But even just thinking about what we can access, okay, a genome is published. How do you take that gene and actually use it in your own chassis or your own organism? You need to convert that into synthetic DNA. And for sake of argument, because again, I think it's top of mind, imagine the US decides to put a tariff, a 100% tariff on synthetic DNA. Do you know actually where you would access it from? Do you know where you could buy that from? And without a domestic supply here in Canada, that would essentially be putting a boot on the neck of Canadians, of the Canadian bioeconomy. So now is absolutely the perfect time to establish world-leading DNA synthesis capacity, just like we did with DNA sequencing. And, I'm going to play devil's advocate here for a moment. So some people will say, well, how do we know that the technology is mature enough? Are we really ready to make an investment in this? And advances in DNA synthesis technology are continuous. And I'm very excited. There's lots of recent literature about enzymatic DNA synthesis as well that may revolutionize things. But there's never going to be a perfect time. I think personally, I believe right now is the perfect time because of the need for this domestic capacity, as well as creating something special that no other nation has made a concerted effort to invest in. So advances are going to be continuous. So why not just invest now? And then we'll reap the benefits, because once there is, even better technology, we will have already developed and retained talent to use it. So just like at DNA sequencing facilities, it requires really dedicated, well-trained individuals. These are people, just like Scott mentioned. It comes down to people, not just equipment, who have full-time jobs to ensure that this capacity is delivered. And by investing in that right now, we ensure that these people are employed, they stay in Canada, and we set up these facilities across Canada so they're accessible and they can act as a node to the Open Bioresearch Alliance. And from an economic point of view, so again, DNA is cheap to replicate and store, but it's expensive to produce. So that actually means that if you have an ability to produce it, you kind of hold, again, the keys to the bioeconomy. And believe it or not, the price of synthetic DNA has been pretty much flat for the last 10 years. Now, this graph is not up to date. It brings us to 2018. But I'm certainly not aware of anything groundbreaking that has made the price of DNA cheaper than 1 cent a base pair. We've been stuck at between 1 and 10 cents a base pair for a better part of 10 years, which is really insane when you look at the price of DNA sequencing, which has just fallen like a rock. And that's where I want to get us to, but that's not going to happen overnight. What that does mean, though, is that even a small reduction in this price, a subsidy, will mean that Canada has an internationally competitive supplier of synthetic DNA. So with just an ability to turn that from 1 cent to 0.8 of a cent or half a cent a base pair, Canada could be the world leader overnight because no one else has cracked that code. No one else is offering it at that price. So it actually means a really targeted investment can make an international impact. And yeah, I'm very excited about it, as you can tell. But like, I think the economic sense is just right here. Okay, so this is kind of what build would look like in Scott and I's minds. So there would be a request stream towards this Open Bio Research Alliance, so where you're requesting new open parts, request for existing parts, a request for DNA synthesis, and the Alliance would output credits for DNA synthesis. So whether it's a project-based or whether you're depositing parts and earning those credits, as well as validated open parts and affordable synthetic DNA. So this is domestic capacity and applying these, or you get credits in return for these open parts that you're depositing. I don't remember what comes out. Hold on. Oh yeah. Right. So rather than those freezers in the desert, well, let's return them to nature. Let's bring them to the bioeconomy and release everything in them. Well, maybe not everything, but certainly anything that's otherwise never going to see the light of day to the bioeconomy in Canada. Yeah, much nicer than freezers in a desert, hopefully. Okay, last little part of this. So test. So this is building to this, but where are these nodes going to be? So it's wonderful to think about, oh yeah, there's all these shiny labs across Canada that are all contributing and everyone's singing songs. It's wonderful. But won't actually storing these things and replicating all those strains in DNA be challenging? So that's where lab automation can play a role here. And lab automation can help with repetitive tasks. So that means replicating plates, DNA assembly, validating parts, just to make sure that the DNA is there and the cells are growing and that everything's hunky-dory. These are not complex experiments, but they do take time. And if we want to think about thousands, hundreds of thousands of different strains or parts, we need robots to help with that. And that brings up this other point, is that lab automation unlocks a scale that's well beyond manual methods. So we can run experiments overnight. We can take time points in the middle of the night. We can miniaturize experiments smaller than what you can manually pipette. And anyway, I can go on and on about lab automation, but they absolutely contribute to testing. So this does require dedicated equipment, but it also requires expertise. You can't just plug in one of these robots and say, build a plasmid and push a big button that says that. I don't know if anyone from my institute's on the call, but like they'll know just how much work that we put in over the last 2 years to get us to this point of, I'd say comparable experiments to what we can perform using manual methods, but using a robot instead. So we're getting closer to essentially replicating what you can do manually. But that still took 2 years of development. So it's a costly investment to do that. So how do we build and sustain this? Now, I'm not ever going to advocate that every lab should have a liquid handler, every lab should have a bioreactor. That's just not realistic, and it's not necessary either. If we're a little bit smarter about how we use this equipment and support this expertise. So we've already, well-funded universities across Canada, and that's in large part thanks to the Canadian Foundation for Innovation. So there's lots of equipment and there's lots of expertise, but it often sits underutilized, and that's just the reality of academic labs. Is that they go on grant cycles. Sometimes they're humming 24/7, but often they're not. And people are getting paid full time to look at a robot and say, wow, it'd be great if I could use that for something cool. So if we don't use it, we lose it. And that includes the talent. So if people are not paid, and if they're not motivated, then they'll move with their feet, or they'll vote with their feet, and they'll leave. They'll find something that is motivating and something that pays more. So the last sort of bit of this idea that we're pitching here is that it's a single request portal for accessing this equipment and expertise. And you can access it just like the Digital Research Alliance using project-based funding. So this means you would submit a grant proposal to the Research Alliance and say, these are all the great things that I'd love to use your lab automation expertise for. And then it would go out to a grant call, and essentially the Alliance would be paying itself to run your experiments for you. You get all of the data, you get all of the resources. The Alliance keeps the staff and the equipment running. So it's win-win in that regard. And of course, that's going to require dedicated money. This isn't money that's just going to appear, or we're not going to do this for free, rather, but it could just appear if there's a serious and willing politician on the call that would be interested. And this model is absolutely inspired by the Agile Biofoundry, and they have a proven track record of what dedicated investment in shared infrastructure can do. And please check that out if you want to learn more. I'm not going to give their pitch, but we could build something like that here in Canada. So that puts all of this together, hopefully, and I think we're just about at time. So there's a project stream now. So you've got a request stream in pink, project stream in blue. So project-based access to high throughput design, build, test, and learn. And based on the success of your application, the wealth of Canadian talent and lab automation and high throughput biology and biomanufacturing is at your disposal. So these will accelerate experiments beyond manual scale, getting to your desired results faster. And I think I've captured these three parts. So this is the storage, so the sharing and distribution of these parts, the building of these parts. So that means synthetic DNA that we're manufacturing here in Canada. And based on your request, what exactly that would be. And if it's open, then you're going to get more credits for making even more DNA. And then, of course, the centralized equipment and expertise. So those are two pieces that you can't have without the other. So equipment and expertise for high throughput design, build, test, learn, and biomanufacturing. I put our emails here. I forget what's next. Oh, yeah. Okay, this is a challenge for you. So if you like this at all, if there's any part of this that resonated, if you've made it this far through the presentation, I actually have an ask of you that we need your help with. So this is Scott and I's vision, but it's not going to go beyond that without your help. So what can you do if you want to contribute now? And unless there's a bag of money that's going to land on my balcony right now, well, actually, let me rephrase that. We shouldn't wait for a bag of money to land on my balcony. There's lots that we can do right now to contribute to this. So as Scott brought up, you need to advocate at your institute for adopting the open MTA. So you're aware of it. You can do your own research. You're all smart and motivated people who are spending your lunchtime listening to us ramble on. So thank you. But now once you hang up on the call, look up what the open MTA is. There's lots of information, and bring that to your tech transfer office and say, can we use this? And can we make the parts that I'm developing in the lab open? What would that take? And they're gonna, they're not gonna like it at first, probably. They might be, hey, I've never heard about that. Tell me more. But it's your job to advocate. You can then also build your own open parts and share them. So ensure that you have institutional agreement to that. You can't be using any institutional resources. I'm going to say that again. You cannot use any institutional resources to build open parts unless you have their full agreement. So don't just do this on a weekend and then say, well, I built a plasmid and it's open. No, you're going to get in trouble for that. That's not the way that grant money can be used unless you have their full agreement. If you do, and if you have institutional buy-in to this open MTA, then you could. Unless, of course, your own have your own independent funding that has fewer strings attached. But for most academic researchers, and certainly for grad students, ensure that you get full buy-in to that and full agreement. And then if you do say, well, I have this plasmid that I built 15 years ago, and I want to share it. Well, you need to do a little bit of homework. You need to ensure that these parts are truly free of patents and MTAs. And that's not always easy, but you can talk to your tech transfer office about that if you're an academic. And if you're at a company, If you have a legal department, you can talk to them about that, or just look up, well, where was the actual originator of this strain or this this plasmid that I built. If you synthesized it by hiring a company and none of those parts have a patent or those patents have expired, then it's likely that you may be able to share it. And then you can also contact us. So there's our emails. Put them up here again. Scott and I would love to hear from you if you're interested in contributing to this. And really, you know, until that bag of money falls on our balcony, I think that we can do a little bit more work to making this a reality. And I think that's it. So I'll leave this up just for sake of conversation, and it is a lot of information. I also mentioned that Scott and I have written a white paper that we've submitted and to an academic journal, but there's lots more detail, and we fleshed out some of these ideas beyond what we could communicate in just an hour. So yeah, thank you.
Thanks, everyone.
Thank you so much for that presentation and just opening up the dialogue to this important work we have to do around building our Canadian bioeconomy. We have a few questions. I think we'll spend maybe 10 minutes for the questions.